Welcome everyone to today's webinar, Successful Migration to a Vancomycin AUC Dosing Protocol, Evaluation, Implementation, and Outcomes in an Acute Care Hospital Setting. On behalf of Becker's Healthcare, thank you for joining us. Before we begin, I'm going to walk through a few quick housekeeping instructions. We will begin today's webinar with a presentation, and we'll have time at the end of the hour for a question and answer session. You can submit any questions you have throughout the webinar by typing them into the Q&A box you see on your screen. Today's session is being recorded and will be available after the event. You can use the same link you used to log into today's webinar to access the recording. If at any time you don't see slides moving or you have trouble with the audio, try refreshing your browser. You can also submit any technical questions into the Q&A box. We are here to help. With that, I am pleased to welcome today's speakers. John Faldaz is a pharmacist by training and is currently InsightRx's Senior Director of Product and Customer Experience. With over a decade of clinical experience in pediatrics and infectious diseases, John now ensures that the InsightRx Nova platform is everything clinicians desire and that clinicians can make full use of all InsightRx Nova has to offer. John received his PharmD degree from Massachusetts College of Pharmacy and Health Sciences and completed PGY2 infectious disease residency training. Craig Randall is a clinical pharmacist at Wentworth Douglas Hospital, a 178-bed community hospital located in Dover, New Hampshire, and also manages their antibiotic stewardship program. Their ASP team has worked on many exciting projects, most recently completing a year-long research project on Vancomycin AUC-based dosing. Craig graduated from University of Rhode Island College of Pharmacy in 2013 and completed PGY1 residency training. John and Craig, thank you for being here today, and I will now pass the floor over to John to begin today's presentation.
Awesome, and thank you, Marcus. Thanks everybody for being here. Excited to get going on this. So as we go through, I'm going to be sort of the bookends on this presentation. I'm going to start with a little background, and then Craig's going to give the meat of why people here with his experience at Wentworth Douglas implementing the Vancomycin AUC dosing. And then I'll come back in afterwards talking about how we can potentially build off that success with some time for questions and answers at the end. So I'll start with the background, and the background is going to be just really getting us on the same page about what we mean by AUC-based dosing and what MAP Bayesian calculators are, one of which is InsightRx. So we'll start with AUC-based dosing. I think most people on the call are probably pretty familiar with the 2020 guidelines for therapeutic monitoring of vancomycin by now, but the guidelines, when they did come out, pretty strongly advocated for AUC-based dosing, saying given the narrow vancomycin AUC range for therapeutic effect and the minimal AKI risk, the most accurate and optimal way to manage vancomycin dosing should be through AUC. So in short, they go through obviously a lot more detail, but they talk about how the true exposure metric for vancomycin is AUC, or area under the concentration curve. Troughs historically had been used as a surrogate because we weren't technically there yet to be able to measure AUC for everybody, but we now are there. And so we're able to measure AC and maintain optimal efficacy and minimize toxicity. They further go on to say that the preferred approach to monitor AUC involves the use of Bayesian software programs. And they go on to discuss proper PK models and a little bit more on that. But they actually don't go into that much detail about what Bayesian software programs are. And InsightRx is one of those. So I can speak to a little more about what we mean when we say Bayesian software programs. Bayesian software actually combines two things: MAP Bayesian forecasting, which we'll talk about in just a couple of slides from here, but also model-informed precision dosing, which Bayesian forecasting is actually a subset of that. When we say model-informed precision dosing, or MIPD, what we mean is it's actually just that. It's using a model, in this case a pharmacokinetic model, to help inform patient dosing and help it be more precise and more accurate. And that concept is not new by any means. We've been doing model-informed precision dosing almost since the advent of pharmacy and dosing. So if you think about things like weight-based dosing and renal adjustment, those are ways that clinicians have decided that a one-size-fits-all doesn't work for everybody, so they've looked to pharmacokinetic models to say, all right, if we use total body weight or an adjusted body weight, we'll be able to get proper exposure for more patients. Or if we adjust based on their renal function, we'll be able to get more accurate dosing for our patients. So it's an interpretation of models, sort of in a rudimentary form. Nomograms had been sort of the big version of that, right? Where you incorporate weight and height and serum creatinine for something like vancomycin to get these bucketed systems for a set of 12 different regimen options for different types of patients. And this is good. It's getting more granular. But of course, as everyone knows, these nomograms are good for a lot of patients, but there are some patients that slip through the cracks that it fails for, mostly because it's a simplification of a pharmacokinetic model. People try to take a pharmacokinetic model and force it into these small boxes. So the next wave of this, the modern form of MIPD, is just to use the pharmacokinetic model itself, to take patient factors like things we've been doing, height, weight, serum creatinine, gender, age, but other things, potentially albumin, race and ethnicity, disease state, all these other things that may play into it that feed into the model and give you something that is precisely designed for this individual patient, as opposed to trying to force it into any type of box. That's the crux of MIPD. And even in this Bayesian software, when we don't have levels back yet, we're using MIPD with pharmacokinetic models to determine our initial dosing. We just use the model, plug all the data in, and say, what does the model say would be the proper dosing for this patient? Now, map Bayesian forecasting, as I alluded to earlier, is used when we have levels in place. And I'm not going to go into the details of map Bayesian forecasting. I'm certainly not a mathematical expert on it. But what it does in a general sense is that rather than interpret levels at their face value, it interprets levels in the context of this pharmacokinetic model. The PK model is what we expect the patient to do. We have a level that came back, but levels have inherent variability as well. So map Bayesian forecasting balances these two things, the model and the level, sort of balances the pull back and forth and comes up with an individualized Bayesian fit, which is shown here in green. And that is shown to reduce sort of the improper pull of bad levels, that variability. It's just, so far in the data, seems to be the best way we can do. The cool thing about it is, as you get more and more levels, each of those levels has its own pull and pulls that Bayesian line toward the levels. And so as you get more and more levels, the pharmacokinetic model becomes less and less relevant because you have more data individualized to this patient, till eventually you can potentially do it based entirely on levels if you have enough levels for the patient. That's sort of in a nutshell how map Bayesian forecasting works. When you put these two things together in software, the benefit that we have are MIPD gives us more accurate initial dosing using the pharmacokinetic model, better than our nomograms can do. We have the ability, because there's software involved, we can convert levels to an exposure metric like AUC very easily. It does all the math in the background. It's a really fancy calculator. We're able to interpret levels from any time point and regardless of dosing history, and that's because these full-fledged Bayesian dosing platforms account for the entirety of the dosing history with each dose, with the amount and the time, and it calculates the residual exposure available for each one. And so you don't have to have a perfect level, a steady state trough, to be able to interpret it. You can interpret any level. You can have missed doses. All those things are applicable. And when it all comes together, the science says that they have improved prediction accuracy in response to levels compared to, just as we said, that flat default interpretation of levels. And when you all put it all together, you end up with therapeutic exposure more quickly and more often in your patients. That's the crux of map Bayesian forecasting and AUC-based dosing. I think that gets us all on the same page. And now I think we can hand off to Craig and his experience. So I'll hand off there now. Take it away, Craig.
All right, awesome. Thanks for that introduction, John. So as mentioned, my name's Craig Randall. I'm the Antibiotic Stewardship Manager at Wentworth Douglas Hospital. I'm going to talk to you about our experience and journey to go live with vancomycin AUC-based dosing from the ground up. So just for perspective, Wentworth Douglas Hospital is a mid to large size community hospital. We're about 200 beds located in Dover, New Hampshire. We went live with vancomycin AUC-based dosing in May 2021, so we've been live for a little over a year now. And we have 30 pharmacists on staff at the hospital who all utilize the program. As John kind of mentioned, background, you guys are all familiar with this. So historically, trough levels have been used as the primary method for vanco monitoring. But although trough levels provide ease of monitoring, recent literature suggests that targeting these troughs of 15 to 20 creates unnecessary risk of nephrotoxicity without improving efficacy. So the consensus guidelines came out, and anytime you can get big organizations like ASHP, IDSA, SIDP on the same page, it's definitely a strong recommendation of best practice. So they now recommend AUC-based monitoring as the primary method for monitoring and treatment of serious infections due to, specifically looked at MRSA, but I'll go into all the populations we use this for AUC monitoring. So historically, calculating AUC has been cumbersome. So until the development of Bayes and PK calculators, you look at the formulas, you'll get a headache trying to do all the math. So they make it a lot easier. Bayesian modeling has effectively generated reliable AUC estimates compared to trough-only sampling. Area AUC to MIC, also known as AUC-24, is now the preferred pharmacokinetic pharmacodynamic measurement to assess vancomycin efficacy. And just because I assume there's a large number of tertiary care facilities on this, I just want to highlight the point that at Wentworth Douglas, about 99.9% of vanco dosing is completed by the pharmacist. So I know there's some systems and hospitals on the call that probably have residents and fellows doing dosing, but we are a pharmacist-managed protocol almost exclusively. So how did we decide and implement and evaluate how to go live? So first thing, our clinical pharmacy team evaluated and researched different pharmacokinetic calculators out there. We looked at the EPIC calculator. So we're on the EPIC EMR. I know there's probably a lot of hospitals that are also utilizing this. We looked at InsideRx, and we also looked at some of the free calculators like DoseMeRx, ClinCalc.com. And ultimately, why we decided to choose InsideRx, probably the biggest reason is that you can use a single level to calculate AUC. You can use a single random level. The EPIC navigator and calculator requires two levels, so it requires a peak and a trough to basically calculate and predict dosing. So that peak and trough, the easiest way to think about it is it's like dosing aminoglycosides with peak and trough monitoring. It's very tedious and cumbersome. So that single level, being able to draw on at an independent time in the dosing interval, is very convenient and very easy logistically for staff. AUC levels can be assessed sooner outside steady state within the first 24 hours of therapy. So we're able to check levels sooner, achieve therapeutic targets faster. There's increased accuracy in special populations such as obesity. There's a Carreno model for patients with a BMI over 40. It's a very easy to use platform. I found it very user friendly. Our staff has felt that it's pretty easy to use. And we have InsideRx has excellent hands-on customer service. You can usually get somebody any time of day with a question or an issue that comes up. Their service team is very responsive. They were great during Go Live, and it was a huge help to us implementing everything. So cost is a big one for a lot of institutions, so I wanted to highlight this. So it does vary by bed size of hospital. So our hospital in particular, we completely offset costs and actually saved money by eliminating our clinical pharmacology subscription and subscribing to InsideRx. So we actually saved money by making this switch. And we actually still have access to clinical pharmacology through pharmacy schools that we precept for. So cost was really not an issue here. Implementation. So basically, we had this idea. We knew we wanted to go live with InsideRx. So first step, we collaborated with other area hospitals to develop a consensus. What are other people doing? And research strategies for protocol development and implementation. We're lucky we're in the Mass General Brigham health system. So there's a wealth of resources, great people to collaborate with. There was other hospitals piloting, testing things out, and we were able to collaborate together to develop a protocol. So then we sat down and developed our own individual vancomycin pharmacist-managed AUC dosing protocol. We obtained approval through Pharmacy and Therapeutics Committee. We navigated the various political channels through the hospital. went through some physician leadership and obtained approval for this process. Physicians, honestly, at this point, they love that pharmacists dose it and they don't have to deal with it. It can be tedious at times, but it's a core pharmacist responsibility. We then trained 3 pharmacists as super users to assist with training and go live, myself included. And basically, we were going to be go-to content experts for people during the training and after go live. For pharmacist training, I'd say with any computer system, it's very hands-on. You have to play with it. You have to use it yourself. So we did a little bit of background. We watched some video modules just for a little background, and we held some Inside RX training sessions, which are Q&A just to figure out general questions, keep people started. And then the core of our training, we created a Vanco competency assessment with patient cases. We had pharmacists actually complete this individually after some basic training. And we did that because we wanted them to figure out where they got stuck, what was confusing, and where their trigger points were. So after that, we then did 1 hour of one-on-one training, pharmacists to super users, so that they could kind of hash out anything they were having trouble with. So everything we do in pharmacy and medicine is evidence-based. So we wanted to see, is this process, it sounds great, but we wanted to look at the effect at our individual institution. So we completed a year-long research study looking at the effect of a pharmacist-driven AUC policy compared to trough-based dosing. And I'm going to kind of go through the research and outcomes on the future slides. So our primary outcome is assessing the percentage of therapeutic troughs pre-protocol compared to therapeutic AUC post-protocol within the time frame of 48 hours. So that's our primary outcome. Secondary outcomes were percentage of Vanco levels drawn appropriately. We had a significant issue with nursing drawing or lab drawing levels inappropriately, leading to redraws, which obviously is an extra patient stick and a cost wasted. Incidence of acute kidney injury, that's probably the biggest reason for the recommendation of this new dosing strategy to reduce AKI, and workflow improvements for staff. We're trying to make things better and improve for staff. So our study design, we're a small community hospital, so it's a retrospective process improvement analysis. We don't have thousands of patients, but this is more realistic to a lot of institutions that are on this call. So we find the data relevant to a lot of hospitals and it's applicable. So we looked at trough-based dosing and AUC-based dosing in 5-month periods. And you'll notice that the dates are a little different with AUC and trough-based. And the reason why is we went live May 3rd, 2021. So we wanted to start collecting data from the ground up. So that's right from go live when we started data collection. We use paper Vanco tracking forms internally at our hospital. This helped us with the retrospective review to flag patients. We use paper tools still just to not lose track of consults because there's a lot of things electronically. Sometimes the physician doesn't order a consult. That helps us with tracking internally. We deem troughs therapeutic on an indication-specific basis. Primarily, the majority were a 15 to 20 goal. And AUC levels were deemed therapeutic if they were between 400 to 600. Our methods, so inclusion criteria, hospitalized patients over 18 years of age being treated for a presumed gram-positive infection. I'll say presumed because as a stewardship manager, I'd say there's a lot of unnecessary Vanco use, but I think that's not unique to our hospital. That's common throughout the country. And we had to have at least one vancomycin level drawn. So if there wasn't a level, we didn't have anything really to analyze. So they were excluded if they didn't have a level drawn. So we had a lot of patients excluded in this time period for no levels being drawn. We didn't look at pediatrics. We didn't look at pregnancy. Anyone undergoing renal replacement therapy, so dialysis or CRRT, were excluded. Acute kidney injury is a big one. So anyone with an acute kidney injury, and really why we excluded this patient population, the guidelines kind of discuss it, but the calculations and population-based kinetics are just not as accurate in that patient population. I wouldn't say that you can't use AUC-based dosing. Sometimes I use it for comparison, but we chose at our institution to exclude AKI. So we use trough-based dosing in those patients. And if Vanco therapy was discontinued before a level was drawn. So this is just a bird's eye overview of our trough-based protocol. Pretty standard. We use a 20 mg per kg load with a 15 mg per kg maintenance dose. You will see on this slide that we used to use and have a pretty large loading dose cap of 4 grams. I will say, with that being mentioned, less than 1% of patients actually got that loading dose. We did a lot of education internally. I just was never a fan of even having the ability to dose that high. So we decreased that, and we followed aligned with the guidelines. So our new max is 3 grams, and our maintenance dose has been decreased from 6 grams per day to 4,500 milligrams per day in divided doses. And that's an initial dosing, which I'll get into later. We did have a special decreasing dosing in obese, renally impaired patients for 10 mg per kg, but that's just a quick snapshot of our trough-based protocol. So our AUC-based protocol, so what happens? So a hospitalist places a CPOE order for pharmacist-managed Vanco dosing protocol, so then we take the reins. So the pharmacist will utilize InsideRx dosing software to determine a Vanco regimen. It's pretty simple. You just enter some basic pharmacokinetic info. John kind of highlighted those characteristics, but age, gender, serum creatinine, body weight, height. And then the dose assist function pops up with a dosing recommendation. So this slide here, there's a lot of info and numbers on the slide. You know, it's distracting to have this much, but I just wanted to highlight, this is what we'll see on our end with an initial dosing after we've inputted this pharmacokinetic data. So in the center, if you focus your attention on the reference table, that's where your dosing recommendations start out from. So you can see the highlighted section recommending 1,500 milligrams every 12 hours, predicting an AUC of 557, which is great, trough of, and within our therapeutic range. So I do want to highlight, and I'm going to get into on the next slide, the reference table is steady state predictions. So this is actually looking 8 doses 4 days into the future based on this regimen. So you have to think about the time that it takes to get to therapeutic AUC. So sometimes pharmacists, we had the issue, would select a regimen that would be in the low 400s within the reference table, and we wouldn't achieve steady state. We'd be subtherapeutic with our dosing. So just to know, this reference table is steady state into the future. So this slide here, I kind of wanted to highlight the difference between that reference table and the exposure table. So I mentioned the reference table is predicted at steady state. The exposure table is looking at real time, where are we now with our current dosing based on the amount of doses given. So I circled after the second dose because we had checked a Vanco level here after dose 2. Our AUC in real time came back at 548 with a trough of 15.7. So we were great. We're therapeutic now. However, if you look above in the highlighted section, this is the great thing about InsideRx. The prediction at steady state in the future in the next few days was going to predict a toxic level. Not toxic, but super therapeutic with an AUC of 718 and a trough of 21. So although we're therapeutic now, on our old protocol, we probably would just let this sit, not check for a few days. But this data tells us we're going to go in a super therapeutic direction. So it allows us to make a dose adjustment and prevent toxicity. So I think a lot of times it's a very dynamic process. You know, you have a real-time view and a future view of what your current dosing looks like. I love that feature, and I think we've caught a lot of toxicity with these features. It's a huge value added to our dosing. So this is what our protocol looks like, you know, as far as indications, how do we dose, and what are we doing? So pharmacists select a regimen. We target an AUC as our primary efficacy endpoint of 400 to 600 for most indications. So you can see that first column there. We use 400 to 600 for the majority of indications. We do use a higher AUC target of 500 to 600 in high priority treatment indications, osteomyelitis, meningitis, endocarditis. There's some absorption issues. We want to make sure we're hitting therapeutic concentrations in those populations. I will say it's a little bit individualized. You know, if we have a bacteremic patient in the ICU, we sometimes take the liberty of targeting an AUC of 500 to 600 because we want to make sure we hit our therapeutic target. For trough monitoring, it's really a secondary target. We're just trying to maintain a trough of at least 10, and that's just to make sure we're not creating any resistance to treatment. So that's our primary efficacy targets. Intermittent hemodialysis, we use pre-dialysis levels. I'll get into that later. And we dose by level or trough-based dosing for AKI or any sort of renal replacement therapy. As far as how do we do levels, so we use random levels as I discussed. So you can do that after at least 2 doses within 24 to 48 hours of therapy. So we usually don't do it after 1 dose just because there's not that much data to interpret. So we usually want at least 2 doses, which is usually within the first 24 hours, so it's not a big deal. And then we monitor serum creatinine and Vanco levels per our protocol. So as far as patient baseline characteristics, roughly equal between groups. So we had roughly an equal distribution of male to female. In our AUC-based arm, we had 256 patients. In our trough-based, we had 200 patients. So we had pretty good numbers for our hospital size for this study. One thing I want to highlight with treatment indication is our distribution of skin and soft tissue infections is very high. So 37.5% of our patients were being treated for skin and soft tissue infection, a lot of cellulitis cases. I'm not thrilled about that as a stewardship manager, using it for uncomplicated cellulitis at times. We obviously are constantly working with hospitalists on this front. So let's get into the data, what happened. So our incidence of therapeutic troughs in our trough-based pre-protocol group, 28.5% of patients achieved their therapeutic target within the first 48 hours. Compare that to post-implantation. So in AUC-based, you can see a huge improvement. So our incidence of therapeutic AUCs jumped to 72.4%. So our levels went from therapeutic from 28% of the time to 72% of the time, which is a huge improvement. I'll get into the specifics of why I think this happened, but there's no question, data doesn't lie, that it's an improved process and it's more accurate. We were thrilled with the results. So one thing I love that Insight RX does is they send you quarterly analytic reports. Lots of data, digs into all the fine details of how your institution is dosing, and really gives you ideas for improvement and where your difficulties are. So currently there's roughly 475 health systems using Insight RX right now. Wentworth Douglas, and this AUC-24 within target range, this is of all levels. So 61% of the time we were therapeutic within that target range. And our target range does increase to a higher point at 48 hours to 72%. And then AKIs, during the first 48 hours of therapy, our AKI rate was 1% for Wentworth Douglas. This is 1 quarter. I'll kind of get into some more data here, but just wanted to highlight this. Kind of getting into some more of the analytics. So this is looking at AUC over time. So if you see that center green section, so this is looking at within the first 24 hours, 24 to 48, and 72%. And it's good. You want to see this getting higher over time. This was in quarter 3, but 72% of our patients were therapeutic at that time frame. And it kind of aligned. It was nice because we did our study independently through our residency program. And it kind of validated the data we collected and showed us that, okay, we're on the same page with our data, and we can kind of back up and definitively, say, what we found in our analysis. So Vanco levels being drawn appropriately, they have a section of how many levels are being drawn. So, and just for comparison's sake, in our trough-based group, about 70% of our levels were drawn appropriately, which is actually higher than some health systems. We have other health systems we work with that are in the 30 to 50% of levels being drawn appropriately range. That increased with the implementation of random levels, less time sensitive, to 97%. So we're not throwing out very many levels anymore. There's still always going to be the one-off cases, but overall, huge improvement. We also, we only check roughly 1.3 levels per treatment course. De-escalation plays a factor, but we had roughly 2.5 levels per treatment course in our trough-based group. I think that speaks to the increased accuracy. Our AKI rates, this is just another slide. This is actually directly from Insight RX, but this is just kind of further highlighting that 1% AKI rate. I will say, when you look at AKI rate, if you looked at our AKI rate pre-protocol, we were probably in the 8 to 10% range. Within this study, we had 11 patients with an AKI and trough-based dosing in this patient population, which was 5.5%. And Insight RX, after going live, we had 1% of patients with AKI. I will say that our AKI rate's in the 5% range. Real time, looking at a year of data, that's kind of where we've kind of landed with our AKI rate. This is just a dosing recap, looking at just large scale. How are we doing with dosing? Where are those levels that aren't in therapeutic range going? So interestingly, our tendency as a team is to underdose vancomycin. So you'll notice that we have 30% of levels roughly that are out of therapeutic range. 28% are subtherapeutic. So our tendency is to underdose. Only 2% or only a few percent of our levels are an AUC over 700. So, and that's really where a trough over 20. So we're doing a great job preventing toxicity, but we're a little hesitant with dosing. This is just a little bit bigger slide just to kind of highlight that data. So you can clearly see our tendency as a team is to underdose. Why do I think that happened? I think it was a new process. I think a lot of pharmacists were a little gun shy with dosing. They wanted to get used to things, kind of get their hands on the process. And we've really been coaching that it's okay to have an AUC over 600, which I'm going to get into. And I think a large portion is pharmacists were selecting a dosing recommendation from the reference table that was barely hitting 400, and it takes days to get to that point. So we're coaching pharmacists to target higher levels initially in that reference table. So as far as workflow improvements, there's multiple workflow improvements that we've had for our staff. So it's less of a burden on our evening and night pharmacist staff. They're really just dealing with new starts. So they're not dealing with levels. They're not dealing with a lot of logistics on the night shift. They love this. We have created an electronic historical database of patients to increase accuracy for subsequent treatments. So what do we do well on a patient? We can replicate that in the future. Or did we struggle? And we can see where our faults were in the past and improve dosing for the future. This is a huge value added, in my opinion. We have pushing 1,000 patients in our database now with a lot of data that we can use for the future. Fewer, less time-sensitive draws. I've kind of got into the benefits there, but the lab loves us for this. It's improved workflow for the lab. We've standardized a lot of our levels. I would say it's a nice to do, not a requirement to standardize those with AM labs, but that's what we try to do because it's more convenient. And increase accuracy of drawing levels, which I kind of mentioned. We're a single, so what are our limitations? It's a single center, retrospective review, small sample size, comparison versus historical cohort. I will say that our data is not currently integrated with Epic. This is probably our biggest pharmacist staff complaint, especially at Go Live. So it's a standalone platform. We have to manually enter data into the system. So that is a little tedious and a little cumbersome. I will say there's a lot of systems that are integrated within InsideRx, and it's really not supposed to be a tedious or hard process. It really just comes down to your IS department and how willing they are to work with you. Unfortunately, we're in the Mass General Brigham, which is like a 15 different hospitals, and IS, there's a lot of politics. Unfortunately, they've been dragging their feet on that end. So we're getting there. We're going to be live and integrated probably later this year, but it's been a 2-year process getting integration, which I think that's abnormal. Smaller health systems that are, they're going to have a much easier time with integration. I think John can speak a little more to that, and we can answer that more in the Q&A session. So how do we improve? So I kind of mentioned that targeting an AUC or having something over 600 is okay. So I think a lot of pharmacists are concerned. We're a little bit OCD in our nature that we want to hit a number perfectly within that range. But just to kind of look at this, an AUC of 400 to 600 is already a more conservative dosing target. It roughly correlates with a trough of 10 to 15. So we've really moved dosing targets from 15 to 20 to 10 to 15 in terms of trough. So it's already more conservative. So it's okay to have an AUC over 600. There's multiple different recommendations in the reference table, which can be confusing. I think pharmacists were selecting an AUC that hits that 400, not realizing that that's at steady state in the future. So we're trying to have pharmacists target at least an initial AUC of 500 to prevent subtherapeutic dosing, especially in high priority treatment indications. How are we monitoring levels? So more levels increases accuracy. So we try to collect a minimum of 2 levels, especially in special populations, poor model fits, or high priority treatment indications. Essentially what we do is we check our initial level. If it's therapeutic, we'll recheck a level within 2 to 3 days. If we have 2 consecutive therapeutic levels, then we decrease our monitoring to once every 7 days. That's provided no acute changes in renal function or anything changes with the patient's status. And big thing is we want to make sure that when we're looking at actual AUC, that it's correlated with an actual random level, because everything else is a prediction. So it's population-based kinetics. So special populations, I want to spend a little time here because I think this is really a huge improvement to our dosing process and really where we've improved our dosing. So within InsideRx, you can create treatment tags. So treatment tags are essentially a free text field. You see the special populations we highlighted. I'm going to go into each one in more detail, but it allows us to fine-tune our dosing in these special populations and figure out how we're doing. Just a quick breakdown of weight. Not unique to our hospital. We have a lot of obese patients. And I think our improvements in dosing in obese patients is probably a huge contributing factor to our increased accuracy of dosing. So low muscle mass, when I say low muscle mass, what do I mean? Generally, patients who have a low serum creatinine at baseline due to muscle mass, low muscle mass, so serum creatinine under 0.5. Let's say you have a patient who comes in with a baseline serum creatinine of 0.28, largely wheelchair-bound, quadriplegic, paraplegic, or somebody with severe musculoskeletal disorder. Sometimes we've had intubated patients who have been bedbound for months. Those patients fall into this criteria. Basically, what happens with the low serum creatinine is it overestimates renal function. So you'll get dosing predictions that are a lot higher than are necessary because of that overestimation in renal function. So we've experimented with rounding serum creatinine a little bit. I will say we've dug into the literature. There's not really a clear established rule for rounding serum creatinine, and kind of the consensus is that it leads to subtherapeutic dosing. That's exactly what happened with us. So only 1 of 8 patients were therapeutic, and we had several that were subtherapeutic. So the bottom line is we tend to underdose in this population, so you have to be really careful with rounding serum creatinine. Building off that a little bit, I will say that you can, as an institution, select a creatinine clearance cap. We chose not to utilize this cap because we have a lot higher population of young, excellent renal function patients that chew through vancomycin, and it led to underdosing in those patients. That's why we chose not to use the cap, but you have that option. Obese, excellent renal function. So this is basically, so we define obesity as weight over 100 kilograms, actual body weight 140% of ideal, or a BMI over 40, with excellent renal function and creatinine clearance over 100. We are doing a great job in this population. So over 80% of our patients are therapeutic in this patient population. That's a huge improvement. Obese, renally impaired, same definition for obesity, just comes down to renal impairment. So you just basically move that dosing interval to what their renal function is. So our marker for that is if they have a creatinine clearance less than 90. It is arbitrary, but this is where patients start to dip into stage 2 kidney disease. We're, again, over 80% therapeutic in this patient population. And then young, excellent renal function. This is by far our biggest patient population. So we have a lot of young, under 40. We have a huge population, unfortunately, of IV drug users with endocarditis. So they chew through vancomycin very rapidly. How we define young, excellent renal function: age less than 55 and creatinine clearance over 100. Again, age is arbitrary. You can kind of discuss that internally in your and within your departments or hospitals. But there's a lot of patients in their 50s that do qualify for Q8-hour dosing. Kind of my marker where I kind of steer away from Q8-hour dosing is when patients are over 60. But that's not a one-size-fits-all, definitely individualized. We're doing a pretty good job, 65% of our patients, and it's because we're utilizing higher Q8-hour interval dosing. This is kind of a key money slide that I would definitely mark down, take a screenshot of. This kind of summarizes our clinical pearls. What do we recommend in each of these special populations? So low muscle mass, I kind of went into, we experimented with serum creatinine. I would say cautiously round to a max of 0.8. Be careful with it. So think about if you have a patient with a baseline serum creatinine of 0.28, if you round to 0.6, you've doubled their creatinine and assumed an AKI for that patient. So you have to be really careful rounding serum creatinine. And basically our take home in this special population, we don't use a dosing interval more frequent than every 12 hours. And I mentioned that because the Inside RX calculator, if you don't have the creatinine clearance cap, we'll recommend regimens Q8 and even Q6, which I would definitely stay away from. Obesity really comes down to the weight-based component. I think that's what was flawed. So basically using less than 10 mg per kg for your dosing weight. So I used a couple examples under obese renal. So if a patient is 150 kilograms, we use roughly 7 mg per kg dosing. If a patient gets as high as 200 kilograms, which is rare, we come down to somewhere in the 4 to 5 mg per kg dosing range. And then the dosing interval just comes down to, do they have great renal function or do they have poor renal function? So if they have great renal function, we keep that Q8-hour dosing. Or if they have poor renal function, we might use Q24 or higher. Young, excellent renal function, I think a lot are more familiar with, but utilizing that Q8-hour interval, and don't be afraid of regimens 1250 to 1500 Q8, especially in patients less than 40 years old. I did want to just quickly touch base on dialysis. So what do we do in dialysis patients? So we utilize for hemodialysis pre-dialysis levels. We draw a pre-dialysis level on the day of dialysis, and then we use an algorithm that's very standard. A lot of institutions use it to dose a post-small dose post-dialysis dose. Peritoneal dialysis, I'd say our volume is extremely low, so we don't have a formal protocol. We basically give a loading dose, and then we check a level within the 48 to 72 hour range. If it's less than 20, we redose at 10 to 15 mg per kg. CRRT and SLED, always a crapshoot. It's always tricky. They're on, off. Sometimes the period is interrupted. But we use random levels, and we redose when Vanco levels are less than 20. So just to kind of wrap things up, this is just kind of an overview slide of everything. So we had an increased amount of levels drawn appropriately. We increased from 70% of our levels being drawn appropriately to 97% after going live with InsightRx and random levels. Huge increase in accuracy. We went from 28% with trough-based dosing to 72% with AUC-based dosing. And I think largely we're preventing a lot of toxicity because we're checking levels sooner, and we're able to see both that real-time and steady-state prediction. And then the downstream benefits are the improved workflows. We have a historical database. We're improving workflow for lab technicians and nursing, and Vanco's less of a burden on evening and night staff. So that's kind of an overview. These are my references. I do want to acknowledge, take a quick highlight of all the people involved in this project. Greatly helpful. They were instrumental in helping go live with this process. And I'm going to hand the torch back over to John to kind of tie things up to show where we're going in the future with precision dosing and some of the exciting things for the future. Thanks.
Yeah, great. Thank you, Craig. And great job as always. We always love hearing these stories. So as Craig said, I'm going to move on, taking this sort of victory that we have in vancomycin in this proven case and talk about how it's more than just vancomycin for us anyway. So we start here with the Bayesian software advantages, with the more accurate initial dosing, being able to convert levels to an desired exposure measure, being able to interpret levels from any point in the dosing curve and regardless of the dosing history, and how map Bayesian forecasting can improve our prediction accuracy along with MIPD, getting us to exposures faster. And obviously that's the end goal and something that we want for many, many drugs. So for us here at InsightRx, vancomycin, as I say, is one drug where this works really well, and we're grateful for those guidelines that the authors felt strongly as well as we do that there's no reason not to move forward with precision dosing for vancomycin. But that's just for us one of many, many drugs where we're involved in doing these things. I'm not going to walk you through all of these by any means, but I do want to focus on just a few and show how the same story exists for these and how we believe that this is where we're moving, sort of the world is moving toward next from what we've seen. And I'll talk about antifungals and specifically voriconazole. I'll mention solid organ transplant and I learned tacrolimus, but you may say tacrolimus or FK506. And then in the inflammatory bowel disease world, I'll mention infliximab, and then not on here, but I'll also talk about anticoagulation and unfractionated heparin. And it's sort of this case that repeats itself where you have these challenges and it becomes pretty clear, at least to us, that Bayesian software can solve a lot of these things. So with the azole antifungals, I think for those familiar with dosing voriconazole, the challenges are right at the top of your head. It's difficult to dose initially because there's patient variability, and that includes pharmacogenetic issues, as well as the nonlinear PK. And that makes it, as you can see, jumping down, difficult to adjust doses as well because of the nonlinear PK. In both of those situations, having a pharmacokinetic model solves that issue, that the patient variability and the nonlinear PK are able to be accounted for by a solid pharmacokinetic model and a pharmacokinetic pharmacogenomic model, which they do exist, incorporating 2C9, 2C19. Next, on voriconazole, some people don't know this, but the actual measure of efficacy for most azole antifungals is AUC, just like vancomycin. It's just that, just like vancomycin, we use troughs historically as a surrogate because AUC has been technically infeasible, but it's just not infeasible anymore. It's very much on the table to be able to calculate AUC directly in an AUC to MIC. So that's allowed now because you can convert the levels to the desired exposure metric. the issue of steady state levels. In vancomycin, we're challenged that we often had to wait 2 days or 3 days for a steady state trough. With voriconazole, it tends to be 5 to 7 or even 10 days, depending on your protocol at your hospital to get the steady state trough. But as you know, with these Bayesian software programs, you can get a level at any time. You can get a level after a single dose if you want to. And what that all adds up to, therapeutic exposures more quickly, faster, and more often for the patients. In solid organ transplant, tacrolimus is the one that I use as my example. I was an ID specialist, so I have only overlapping experience with SOT. But what I saw in the Children's Hospital, where I worked for a long time, was that in the initial dosing phase, when they're coming just off of transplant and they're on IV, we know that it takes a certain amount of time to get to steady state, but we're sort of impatient to get there. We want to leapfrog and get up there. So we end up doing a lot of frequent sampling in the beginning and trying to sort of extrapolate out or overshoot, undershoot, and then balance out. And we end up kind of doing a back and forth. Whereas if you have Bayesian software, that single level you get is extrapolated out to a steady state value for you right away. You can figure out the adjustment you want to make, including a load or a mini load if you want to, and just pop them right up to where they need to be off the bat and convert them to oral treatment just as easily. And then similarly for tacrolimus as well. Tacrolimus is AUC-based. If any of you have seen the consensus document that was released in 2019 for tacrolimus dosing, they stopped just shy of recommending AUC-based dosing. And reading between the lines, it seemed to be that they knew it was the right way to go, but they felt the technology wasn't there yet in 2019. And of course, the vancomycin guidelines came out in 2020 and said, technology's there, there's no reason not to do it. And so more and more we're seeing people saying, why not just measure AUC with tacrolimus rather than having to use a surrogate of trough, which you can do, obviously, with the Bayesian software. And then in the outpatient world, there's issues of adherence, missing doses, taking drug holidays, if you will, or sort of logistical convenience in terms of doing things, being able to get a level at the right time. Oftentimes, if you don't get a level time perfectly, it can give you misleading results, or it can potentially be useless and you just have to wait till the next visit. And with the ability to interpret levels at any time, again, you're right there. You can get your therapeutic exposures faster, more often, stay in range. Same story with infliximab. Infliximab is interesting because, at least in my experience, inflammatory bowel disease was not really the purview of pharmacy. It was handled largely by gastroenterology and largely in the outpatient realm. But with this increasing impact that we've seen of TDM and therapeutic drug monitoring, pharmacy is beginning to take a larger role in infliximab again and create that same partnership that we have with many other drugs. If you're less familiar with infliximab, these monoclonal antibodies are dosed every 6 to 8 weeks. And so very often the initial dosing is subtherapeutic because of patient variability, which of course is solved by having a good PK model that you can just give better dosing from the onset. And then when it comes to getting the steady state level, that is 8 to 14 weeks. So you're talking 8 to 14 weeks of potentially subtherapeutic exposure before we can make an adjustment and then wait another 8 to 14 weeks historically. Whereas with software like this, you can give a single dose, get your sample after that, and then adjust as needed. There's also, not written here, but the challenge in IBD and the monoclonal antibodies for anti-drug antibodies that can develop, and they tend to develop more frequently with subtherapeutic exposure. It's sort of almost like that homeopathic effect where you have just enough to cause, or like a resistance effect with antibiotics, where you have just enough drug to spawn the resistance to it. And the thought is that by being able to give better dosing up front, data seems to show that you can reduce the risk of anti-drug antibodies and reduce the risk of having to switch to a different and potentially more expensive drug or risk extended prolonged morbidity for our patients. Again, therapeutic exposures are faster and more often. It's a broken record. Lastly, with heparin in the anticoagulation space, this is admittedly not pharmacokinetic because we don't measure heparin levels. This is a PK/PD model incorporating pharmacodynamics because we measure APTT and AT10A. A lot of the same story. When you talk about initial dosing, that could be helped by a model. I think a lot of people struggle, especially with CAPS and obese patients, some of those less standard or higher BMI patients. It can be tough to figure out what the initial dose should be, and a model would help perfectly with that. And then the struggles with sampling with many of us have protocols that involve nursing or even with pharmacy that have these very tightly regimented protocols that have Q6-hour sampling and adjustment that have to you have to time your sampling, the sample has to come back and you have to make your adjustment and then get a sample again. And it's very easy for things to slip out of order. But the flexibility of being able to draw levels at any time and make better adjustments, including, again, mini loading doses or holding a dose for a while and having more awareness about when an exposure metric will get to the place we want it to be, allows more flexibility and prevents a lot of those errors because we can interpret any level, and that means it allows us to order levels at any time and also interpret levels that may not have been drawn optimally. Over and under coagulation with suboptimal dose adjustments, again, when you have the model and you have map Bayesian forecasting, you get a better idea and you know what to do. And in the end, including co-managing ATTD and APTT, we can have shared modules that have both of these things in there. It can be managed in one single place without having to flip all over the place in multiple parts of a chart to figure out where all your data is. All in one space ends up with, again, therapeutic exposures faster and more often. You can see how this sort of repeats itself over and over again across different areas. I put up this one slide, and I want to make sure we have time for questions, just to talk about briefly a differentiation of InsightRx versus potentially other people in the Bayesian software space. And there were a few questions about this. So at InsightRx, we believe in this end-to-end solution of precision dosing intelligence and a cycle of continual improvement that starts with this dosing decision support tool that Craig talked about, InsightRx Nova, where we're using the best available data and the best available models to dose our patients. But that's just the beginning, because that data then feeds into a real-time analytics platform. And I saw some questions on this. We can drill down into the results and find out where we're doing well, where we can improve. A lot of the stuff that Craig showed you was from the Apollo dashboard and the reporting that comes out of it for real-time analytics. And it allows us to see some of these subpopulations where we might be able to use a different model, or potentially, as we go down to the continuous learning piece, which is less a product and more a process, our data science team and our clinical team of PharmDs come together and look at the data and say, hey, there may be room to analyze this data on a larger scale, figure out which models are optimal, or potentially even iterate on the model and come out with an improvement, which we've done a few times so far, once in terms of serum creatinine rounding that Craig actually talked about, and once in terms of EGFR capping, where we found that some models benefit from it and some don't. And when you're done with that, all that feeds back into dosing decision support. Oftentimes 6 to 9 months ahead of publication, just because of how long it takes to publish. And so now your best available data is now being iterated over and over again and feeds into this cycle of continually improving, that we have better and better ability to dose month after month and year after year. All right, you can tell I get super excited when I talk about that stuff. I come back to this slide just to show all the exciting opportunity there is and where this process can be repeated. I do want to leave some time to talk for questions, so I'll come over now to the question and answer session, and I'm guessing Craig's going to pop up and join me as well, and we can answer a few of your questions.
Absolutely.
All right, yeah, we'll go ahead and get started. Time for a few questions. First off, I wanted to answer something a couple of audience members asked. You will be able to get the full recording of the presentation today with the same link you used to log in once the webinar is finished. But with that, first question we have is for Craig. How difficult was it for your pharmacists to migrate to this new type of dosing?
I'd say, change is always hard. I'd say we definitely had some pharmacists who were supportive. We had some who were more resistant to change, who had been doing trough-based dosing for 10, 15, 20 years. So with anything, change is hard. It's a different process for sure. I'll say it's more dynamic. It really looks at total Vanco exposure in a 24-hour period. But over time, data really doesn't lie. We've clearly increased our accuracy and efficiency with dosing. So honestly, at this point, now that everyone's used to it, pharmacists are very happy with the process. It's really working really well. It took 6 months to hash out all the details. There was definitely some difficulty that happened. But we had really, being in a smaller hospital with 30 pharmacists, we really have good, close working relationships with everyone on our team. And we're able to get that education to them and help with anything that they need. So Go live was definitely busy, but we're doing great now.
Okay, excellent. Looks like next one is for Craig as well. Can you explain how you used treatment tags?
Sure. So treatment tags, as I kind of mentioned, it's a free text field. So anything that your hospital wants to delve into further, you can free text a custom treatment tag. So we picked those individually at our hospital. But I noticed some questions about amputees or patients with very specific pharmacokinetics. You can create a treatment tag if you have a patient who fits those. I will say in some of those funky or special populations, like I mentioned, the low muscle mass kind of fits that criteria. The calculator I would take with a grain of salt. It does get a little bit tricky with those low serum creatinine values. So just definitely take that into consideration. But it's great for analyzing those specific patient populations you want to kind of dig into further. It makes for great residency projects to kind of dig into those. Selecting a treatment tag is not automatic. It's a manual selection that pharmacists or whoever's using the software has to select. So that is, one limitation of that, but it's very useful. We found great benefit from that.
Perfect. Thank you, Craig. All right, John, we'll give you a chance on this one. Can you talk in more detail about the integration options?
Sure. So normally integration with us is an embedded integration. So InsightRx appears sort of, if you're in Epic, as it's an activity tab. If you're in Cerner, I'm blanking on the term, but it sort of appears in place of your chart as an embedded window, and all the data that's needed that would be manually entered, meaning all the patient information, all the dosing information, the levels, the labs, are all imported automatically from the chart into InsightRx as soon as you open InsightRx from within your chart. So it feels like it's a part of the EHR that you're in, and you interact with it when you close InsightRx. You just keep on working with your chart, and that's with sort of all the major chart systems, as well as a partnership with Theradoc and Premier.
Okay, perfect. This next one's a toss-up. When a patient leaves the hospital with a PICC line and an order for Vanco for example, 4 weeks, how are safety labs drawn by the VNA? Can the VNA draw an AUC, or do they still need to draw a trough?
Do you want me to take that one, John?
Yeah, go ahead. I'd love to hear your experience on that.
So, I definitely say the outpatient experience is definitely a limitation because a lot of other outside facilities don't have Bayesian software. So we typically do go to trough-based dosing. However, that being said, when we have a patient being discharged on long-term vancomycin, there's different ways. You're looking at total vanco exposure. If we can create a Q24-hour interval that works for a patient, we can do that. So sometimes we will create an outpatient regimen. It allows you to kind of, with those predictions into the future, look at how an outpatient view would look, but they are using trough sampling on the outpatient side. That being said, could you use it in the outpatient setting? That's probably an improvement for the future. I think we definitely could. Probably what that would entail is we'd have to have, someone who's honestly probably coordinating that treatment, either our ID physician or myself, getting those levels called back in, because you can draw a random level, look at the time that the dose was administered, and calculate an AUC and a trough secondary to that. So it's definitely possible. It's just logistically tricky when we have several different outpatient facilities and VNAs. So they're using the traditional trough-based methods primarily right now.
Perfect. All right, likely the last one we'll have time for. Are you using AUC-based dosing for all indications or only MRSA infections?
All indications. You dig into the data, they look at multiple different patient populations. It's strongest in MRSA. But you also got to keep in mind a lot of treatment is empiric. We don't necessarily have a defined pathogen, but we are using it in all treatment indications. We haven't found, I saw a lot of questions about length of stay, treatment efficacy. We haven't seen any difference as far as increase in treatment failure or increased length of stay. If anything, we've improved length of stay with our reduction in AKIs. So it's not causing any harm from an efficacy standpoint. And if you think about it, think about an Enterococcus UTI versus MRSA bacteremia. If you've proved efficacy in MRSA bacteremia and endocarditis, you've probably maintained efficacy in enterococcal UTI. So we definitely have used it in all treatment indications and had no issues.
All right, perfect. Well, yes, that is all the time we have for today. I want to thank John and Craig for an excellent presentation and Insight RX for sponsoring today's webinar, as well as the audience for great engagement here. To learn more about the content we presented today, please check out the resources section on your webinar console and fill out the post-webinar survey. Thank you for joining us today, and we hope you have a wonderful afternoon.